Hi friends, so now I'm back with my next video, and in this video we are going to talk about ELISA, which is also known as Enzyme-Linked Immunosorbent Assay. So this is a very important technique which is used in biochemistry as well as in immunology to detect and quantify the presence of a particular protein, which can be an antigen of a virus sample, the bacterial sample, or it can be like a receptor or the antibody specific for that antigen present. So I will talk about how this technique work and what is the principle behind it and different type of ELISAs which are present. So please make sure to like, share and subscribe my channel to get more videos related to this. So, what is an ELISA? As I have already talked about, it is a biochemical technique which is used in immunology to detect the presence of an antigen or antibody in a sample. So, what does that mean? Suppose you have I will go with the examples and then I will explain how it works. So, if you know the pregnancy test kit, so the test kit, if if a woman is pregnant, you will see that in the pregnancy test kit you will get two lines. But if the woman is not pregnant, then there is only one line. What does that mean, and how does it work? So this test kit is based on an ELISA. So when a woman is pregnant, there is a particular protein which is formed only during the pregnancy, which is called HCG. And when that protein is present and the sample is loaded onto the kit, it will move. And then the test kit, they have the antibodies against this protein. So the antibodies only bind to a particular antigen, like they have the specificity for a particular protein or a particular antigen. So in the pregnancy test kit, the antibody they are specific for the protein HCG, which is only formed during pregnancy. So if a woman is pregnant and you pass the sample through it, the antigen, the protein that is present, it will bind to the antibody and then you see a line. But during if the woman is not pregnant, that protein is not present, so the antibody they have no protein to react with, so we see no line, like the line which is for the test. And the same principle is used for the COVID-19 antigen test kit, the rapid test kit that show you the control line always, which means that it's working, and the test line, which will be only positive, like you will see the second line only if you have the COVID-19, because the antibodies that are present, it binds to one of the protein of COVID, SARS-CoV-2. The same is used for the diagnosis of HIV and measurement of cytokines as well. So now I think the principle, like the function, how it works is clear, that it detects the presence of protein by the antibody in the test kit, or it can be vice versa as well. So this is a very important technique and I will talk about the principle behind it. So the basic principle that it works with, as I have told you, that in the test kits you have antibodies already present in the sample, like present in the kit. So this technique we generally do in the lab in the microplates, 96 well plates. So in the wells, you have a in the wells are already coated with the antibody. So suppose you want to identify the protein in the COVID-19, so you already immobilize, like you have a plate, you put the antibodies and you let it the antibody to bind to the plate. That is called the pre-coated antibody pre-coated antibody plates. So suppose you have a well in which the antibody is already coated, and then you add the sample in which you want to check the presence or absence of a protein. And if the protein is present, as you can see here in the red, the protein will go and bind to the antibody, and the rest of the protein, which are not specific for this antibody, will not bind to the antibody. So now the antibody binds to the antigen, and now we want to detect. And how can this can be detected is using the different antibody which is again specific for the antigen. So you can see we have one antibody which is already coated, and then we put the sample that we want to test if the protein is present or not, and if it is present, we put another antibody which will go and binds to the antigen which is now present with the different antibody. And then there are different steps, so there are different type of ELISA. Some uses this principle, and in some it uses the same principle like the antigen antibody binding, but then with different reactions and what they use. And in this reaction specifically, we put an another antibody which is not very specific. So you see, so in the antibody, this region at the top you see this is specific for every antigen. So if the antigen is not present, it will not bind here. But the next portion that you see, the long at the tail at the end, this and most of the antibody, this is called the constant region. So this is not very specific because each antibody has a Fc region which is the same. So we put another antibody which binds to this constant region which does not change from antibody to antibody, and then this antibody is linked with one of the substrate, right? Sorry, with the enzyme. So we have the first antibody which is immobilized, and then antigen, and then the antibody which is specific for the antigen, and then we put another antibody which is called the secondary antibody, which is not very specific because it just binds to this Fc region which is same for different antibodies. And then this antibody is linked with an enzyme, and when we put the substrate, which is called the TMB substrate, the enzyme will react on this substrate and form the color product. And this color we can detect by the absorbance. So only if this antigen is present, then only this complex is formed and we see the color. If this antigen is not present, there is no colored form. And then we can also detect the amount of antigen present, because the higher the amount, the more color you see, and then the more absorbance. So this is the basic principle behind the ELISA. So now we will talk about different kinds of ELISA. So there are basically three different types of ELISA: direct ELISA, indirect ELISA, and sandwich ELISA. So I will talk in brief about all three different type of ELISA. So first we will talk about the direct ELISA. So as the name suggests, this ELISA is very direct. So what you have is you have the well, you coat the well with the antigen of your interest. So suppose you have a sample and then you purify from that the antigen, the protein that you want, and you want to detect how much protein is there or not. So you put the antigen. So instead of like having the plate which is pre-coated with the antibody, now you have a plate in which you coat the antigen. You immobilize the antigen and then you put the antibody. So before, as you have seen in the principle, you put the antibody which is specific for the antigen, and then you put another antibody which contains the enzyme. So in this, everything is done in one reaction. That's why it is called the direct ELISA. So then you have the antibody which is specific for the antigen, and then the same antibody is linked with an enzyme. So you don't need a third antibody which is like linked with the enzyme. So in this, it's very direct. You have only one antibody which is linked with enzyme, and then if you put substrate and the antigen, like the antibody binds to the antigen, you will see a colored product. So as you can see, this is very simple. It's only three step, and then you don't need multiple antibodies. That's why it is called the direct ELISA. But the problem here is, um, so the advantages is it's very simple. You don't need so much reactions. It's less time consuming. But then there are disadvantages. First thing is it's a very expensive, because then you need all the primary antibodies linked to an enzyme. Like you need, if you are working with like three different protein, then you need three different enzyme which is conjugated with an enzyme. So like getting this specified enzyme, conjugated enzymes, the antibody specific antibodies with the enzyme is very expensive. Then the one in which you have the secondary antibody, which is not very specific. So it's expensive. And then because as you see in the previous one, it forms a proper complex, so the signal is amplified, but in this case, the signal is not amplified that much. That's why to improve this, we have the indirect ELISA. So what we do in indirect ELISA is first step is the same, that the antigen is coated, like you immobilize the well with the antigen of your interest after purification, and then you put an antibody, but the primary antibody which is specific for the antigen. But in this case, this primary antibody is not linked with any enzyme. So as you can see here, this antibody is not linked with any enzyme as in case of the direct ELISA. So we are saved with the money, so it's not that expensive now. And then we put the third enzyme, which is not very specific. And if you can see in this, if there was no secondary antibody, only the primary antibody is linked with the enzyme, you will see only one enzyme here. But now the simple is the signal is amplified. As you can see, two or three antibody can bind here, and when we put the substrate, we get lot of color from here, from here. So it's not that expensive, and then you get the signal amplified. So indirect ELISA is better than the direct ELISA, but then there are specific things that the secondary antibody that we have, it can be cross-reactive and it can bind to the non-specific places as well. So to improve this, what we have is called sandwich ELISA. So sandwich ELISA is the most common technique which is used in the different research labs, and I think this is the most widely used rather than the direct and indirect ELISA. So the principle is mostly the same, which we have talked about before. So instead of immobilizing the antigen as the direct and indirect ELISA, in this the plate is coated with the antibody, which is specific for a particular antigen. And then you block the non-specific antibody with the blocking buffer, and then you put the antigen of your interest, and if the antigen is present in a sample, you will get the antigen binding to the antibody, and then you put the biotinide label, the or like the antibody which is specific for this particular antigen, and in this case uh, we have the antigen and then the second and another antibody which is specific. So now you get two specificities, one before the binding of antigen and then another specificity. So this reaction is very, very, very sensitive. And then we put another antibody, the secondary antibody, which can be streptavidin HRP or which can be any other secondary antibody like we have talked about. And then we put the substrate, right? Because now we have put the enzyme which is tagged with an enzyme. And then if we put the substrate, then if the protein is present and this complex is formed, the substrate will form the product which is in color. So now we can identify the color of the reaction, right? So I think it's clear how different type of ELISAs work, and which is most common is sandwich ELISA, which is used in different research lab as well. It is the most common technique used worldwide. And if you have any doubts, please let me know in the comment section, and please also let me know other topics that you want to learn more about. And I hope you like the video, and please like, share, and subscribe my channel. Thank you.